All right, ladies and gentlemen, today we are going to be talking about DNA. Yes, DNA. What is DNA? Well, DNA is this, right? That's it, a flash drive. It is an information storage device inside of each one of your cells. That's right. You have information storage in your cells. Come along over here. And remember, you guys all drew a cell, and inside that cell, you have the nucleus. And inside each nucleus of every cell, you have DNA. That is the nucleus's job, to hold DNA. And just like this device, DNA is going to hold information. Now, what information? How to build you. That's right, guys. You actually need to know how to build yourself. It may not be, knowledge up here, but it is information that you need inside of your cells. Parts of your cells are going to read that information, and other parts of your cells are going to assemble you. So it needs the blueprint to build you. So let's take a quick look at DNA. So we're going to do a really quick rundown, big picture idea before we jump into the actual notes. You are used to seeing DNA look like this, if you've ever seen it before at all. This is not what it really looks like. Right? This is a drawn picture. And actual DNA is much more difficult. It's much more compressed in between bends, so it's really hard to see. When we draw it, we stretch it out so we can see details. But there's more detail than that. Gonna look a bit more like this, ladies and gentlemen. Let me bring you in closer for a close view. That's right. These bars going across that you're used to seeing, those are all molecules, right? And the stuff going down are also molecules. This is a big molecule made up of smaller molecules. Oh, am I remembering something from chapter 2? Oh, that's unfair. Yes, polymers, or in this case, a macromolecule. You remember macromolecules, big polymers made up of tiny little small monomers? Big polymer, monomers that make it up. Here's the monomer. It is called a nucleotide. This nucleotide is made up of three things that we care about right now: a phosphate, a sugar, and a base. Phosphate, sugar, and a base. The sugar is known as deoxyribose, and the base is a type of nitrogenous base, meaning nitrogen is involved. That's all you care about, why we call it that, all right? So we take this phosphate, sugar, and base, and we attach it to another phosphate and sugar, and another phosphate, sugar, another phosphate, sugar, all the way down. And they hold on to each other really tight. And same thing on this side. This base hooks onto another base. But look. They only come like this, where the phosphate's on this side and the base is on this side. So if you want the base on this side, you got to flip the whole thing upside down. So now we have an upside down phosphate sugar base. And then the bases are attached to each other, and the phosphates are attached to the sugar, and they will go down. And as they go down, there will be a twist. That's what you're used to seeing here, the twists. So this is actually looking at them from the side. I twisted it, and they twist around completely. So the right side up nucleotides are now on this side, and the upside down nucleotides are now on this side. Now, why is that all important? Because it is a message. I said this is information, right? We are talking about letters of an alphabet, right? These bases are all letters of an alphabet, and they come in 4 letters. That's right, guys, an entire alphabet made up of 4 letters. These 4 letters are adenine, thymine, guanine, and cytosine. Let's shorten that up. A, T, C, and G. That's what we were going to call these letters, as these bases. A, T, C, and G. Adenine, thymine, cytosine, and guanine. I actually had guanine here and cytosine here. Not that that matters, but these are the letters of your DNA alphabet. And just like the letters of the English alphabet, if we rearrange them certain ways, we can make messages, right? I can take the letter C, and by itself is nothing. If I put it next to a letter A and put that next to the letter T, C-A-T, cat. Now I have a word. Same thing here. If I put these letters together in certain orders, I make messages. Not just words, but full sentences, full instructions, how to do something, how to build you. And this is a message. What does this say? I have no idea because I don't speak nucleotide, but it is a language, it is a code, it says a thing, and that thing is something having to do with how to build you. We will go into the building of you later, but let's actually go take notes on the DNA itself. This word is deoxyribonucleic acid. That is a huge word. So we're just going to say DNA, deoxyribonucleic acid. But first, DNA structure. DNA is what we know as an organic compound made of repeating units called nucleotides. That's a fancy way of saying what I just said, right? You get a bunch of nucleotides together and you make a nucleic acid, DNA. DNA is going to store and transmit genetic instructions for cells to make proteins. Guys, when I was on the whiteboard, I said that the DNA tells you how to build you. But think back to chapter 2 again. It's going to keep biting you in the butt if you don't remember chapter 2. Proteins are what you are made out of. Your body is all proteins. So DNA tells us how to make those proteins and where to put those proteins and how to put them all these proteins together to make your body. That's what we mean by genetic instructions. Now, stores it because DNA is basically a flash drive, so it's going to store that information. Transmits, well, you got to have kids somehow. You are going to transmit or send your genetic information to your child, so that your child will look like you, or at least partially like you. And each molecule is going to be made of 2 long chains of nucleotides. This, 2 chains, right? This is a nucleotide, and here you have a chain of nucleotides, and here's another chain of nucleotides, and they're attached to each other. And just like we saw on the board, those nucleotides are going to be made of 3 parts. That deoxyribose sugar I told you about, the phosphate, which was that circle, and the nitrogen base, or as I like to call it, the nitrogenous base. Same thing. And we got 4 of those bases, as we said on the board: adenine, guanine, thymine, and cytosine. Adenine, guanine, thymine, and cytosine. Remember those words. Here is that double chain. And because it's bending, there's a fancy word for this bend. It's called a helix. So there's double chains. We call it a double helix. Just like we saw on the board, the chains are made up of those nucleotides, the phosphate sugar base. Phosphate sugar base, phosphate sugar base. Taking a look, zooming in, you see the phosphate and the sugar and the base. Look at my mouse moving around here. Phosphate, sugar, base. Phosphate, sugar, base. And this phosphate is attached to this sugar, this phosphate, this sugar. So these are attached this way, and these 2 are attached to each other. And then these are attached this way. So it's a whole bunch of attachments. What you have to care about is this attachment, the phosphate and the sugar, and this attachment, the base and the base. Those 2 attachments are what you care about. And this is going to be a bit more accurate thing to look at. This is the molecule, broken down by carbon. Remember, these are organic molecules, and we learned in chapter 2 that organic molecules all have carbon in it. So each number represents a carbon. And there are 5 carbons that we care about. And we name them prime. That's what the little dash right here, the apostrophe, is called prime. So 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. And that is how we name these sides, right? This is the 5 prime to 3 prime direction, because here, the 5 prime's on the phosphate, and then you go down, and the 3 prime is there in this corner. That's the 5 prime here, and the 3 prime here, and that 3 attaches to that 5, and then from 5 to 3. So we go 5 to 3, 5 to 3, 5 is the 5 prime, 3 prime side. This side's upside down, so we call it the 3 prime, the 5 prime side. That will become important in your next set of notes when we talk about how to make DNA. So hold that in your mind? Going back to those 4 bases, they are separated by structure, meaning what they look like, right? They're not just rectangles. That's just my lazy way of drawing them, all right? They actually have a very specific shape. And there's 4 of them, so there are 4 specific shapes that you need to know. No, you don't have to memorize them, but you have to have an idea of them. Adenine and guanine are what we know as purines. Thymine and cytosine are what we know as pyrimidines. Why are they called that? I don't know. I didn't get to name them, but know them for the test. So we got adenine here and thymine here. Adenine was the purine, thymine was the pyrimidine. There's a lot of words. I will say that slowly for you. Take a look here. There's a hexagon shape, 1, 2, 3, 4, 5, 6 sides, hexagon, and then a pentagon, 1, 2, 3, 4, 5. That is 2 agons touching each other. Over here, there's only 1, just the hexagon. That's it. So this is a bigger molecule, and this is a smaller molecule. This is how I remember them, and this is really stupid. People remember stupid things. Adenine is bigger. The bigger molecule gets the smaller word, purine. Pyrimidine is the bigger word, but it has the smaller molecules. Weird, right? Adenine is a bigger molecule described by the smaller word. Thymine is the smaller molecule described by the bigger word. And here we see same thing, guanine and cytosine. Guanine is the purine. Hexagon and pentagon, 2 agons. I don't know what to call them. Geometric shapes, that's what we'll call them. So this has 2 geometric shapes, and this only has 1. And here's the important thing. If you're only going to remember one thing today, this is what you remember. Adenine and thymine attach to each other with hydrogen bonds. The dotted lines are hydrogen bonds. Cytosine and guanine attach to each other with hydrogen bonds. Always. That is the rule. Adenine and thymine are always together. Cytosine and guanine are always together. And they get the hydrogen bonds to hold them in place. And that's what we see happening in my DNA drawing over here. Check it out. Adenines with thymines. Cytosine with guanine. Guanine with cytosine. C and G's, A's and T's, A's and T's, C's and G's, C's and G's, A's and T's, A's and T's, A's and T's, A's and T's, C's and G's. They pair up like that every single time. If there is an A, there's a T with it. If there's a C, there's a G with it. That's all you got to remember. And there are 2 good ways to remember this. A lot of teachers like to tell you A with T because apples grow in trees. And C's and G's because cars go in the garage. And that's one way. What I like to remember is A's and T's are both made with straight lines. C's and G's are made with curved lines. They look similar. That helps me. Whichever one works best for you. A's with T's, C's with G's. Just like in the picture, they form a double helix, 2 chains wrapped around each other like a spiral staircase, because these A's and T's don't fit together like perfect puzzle pieces. The C's and G's don't fit together perfectly. When they get together, they got to kind of bend into place. So when they lock into place, they have to bend in to lock in, and that's going to make the whole thing twist a bit. And then as they lock in, they each bend and twist a little bit more until, I think, like every 10, I didn't actually count this one out, but every 10, 10 and a half, they bend all the way around. And that's how you get the double helix shape. When they're attaching, the phosphates and sugars are held together. You've seen that? I don't need to show you again right now. They are held together by what we call a covalent bond. Remember covalent bonds where 2 atoms are sharing electrons? And they don't want to let go of electrons. So this is going to be a really super strong bond. They're holding on really tight to that electron. So the phosphates and sugars are holding on really tight to each other. It is really difficult to break these. These are the strongest bonds in biology. The bases, on the other hand, the ones in the middle, the adenines and thymines, the cytosines and guanines, are held together by hydrogen bonds, which are very weak. Where covalent bonds are sharing electrons, physically touching, physically holding onto each other, hydrogen bonds are not physically sharing electrons. Instead, they're more like magnets, where there's a positive side on one side and a negative side on the other end, and just like magnets, they're drawn to each other, but they're not physically holding on, so it's very easy to break them. Just like chapter 1 with water molecules being cohesive. Water molecules stick to water molecules. It's very easy to break that bond. This is good. We want to break this bond. We'll learn about why that's important in other sets of notes, but we need to be able to break this bond. And as I said, the lateral twist every 10.5 base pairs. Here you go. I wrote it again up here. The lateral twist every 10.5 base pairs. So after about 10 of these, and then a half, because it's in the middle. So it's half on this side, half on this side. And I did say that in reality, these are twisted much more tightly together, right? They're spread out more in animated pictures so that we can actually see them better. But this is what it really looks like. That's right. Someone actually managed to take a picture of DNA. This was very difficult, by the way. It took the perfect conditions. You had to have the world's most powerful microscope called a scanning electron microscope. You had to have the absolute perfect conditions in the room, with the DNA, with the setup. Everything had to align perfectly. And look, it doesn't look like the twisted ladder you're used to seeing. It looks more like a screw. Here's zoomed in even more, and this is where all the twists. So you actually have about 10 bases, 10.5 bases between here and here. And this is what your DNA is going to look like. All right, the base pairing rules, which I've already talked about, so I'm going to breeze through it quickly so we can get it written down in your notes. Cytosine always bonds with guanine. Adenine always bonds with thymine. There, nice and simple. So you probably have an assignment. At the time of filming this, I didn't make any assignments for you on this chapter yet, so take a look at Google Classroom and see if there's anything there, but I will probably make this a Google drawing where you have DNA and you have a cytosine. So what goes with a cytosine? A guanine. All right, and then on a Google Doc or Google Drawing, probably, I'm going to ask you to fill in the rest of these. Guanine goes with what? G goes with what? A goes with what? T goes with what? All the way down. So look for that. Now, there is a rule, and this is a rule that you guys need to understand. This is a rule that's going to come along with some math. Oh, no, not math. That's right, guys. Math. I'm going to ask you to do math in science. What kind of monster am I? Yes, this is easy math, though. Yes, the easiest math you will ever do in this school. I promise you. All right, so check this out. We talked about this in the vocab, I believe. Because they're in pairs, there is just as many adenine as there is thymine. And there's just as much cytosine as there is guanine. I think the example I gave in the vocab were shoes, right? Because shoes come in pairs, you have equal numbers left and right. If you have 3 pairs of shoes, then you have 3 left shoes, adenine. Then you also have 3 right shoes, thymine. Simple understanding there. Because Dr. Chargaff came up with this idea, we can do a little bit of math. You see, instead of trying to count out every single A and every single T and every single C and every single G in a strand of DNA, which would take forever, with this idea, we only have to count out one of these. So if you remember percentages, 100% means all of them, right? All of them. So really, really simple math for you. I'm going to let you already know that 10% of the DNA is adenine. So right here. No, not your DNA. I don't know what your DNA looks like. This is some pretend patient that we are working with, all right? And in this person's DNA, 10% of it is adenine. So we can go ahead and put 10% right there under A. Before we go into any other numbers, let's organize this a bit, all right? Boom. Where I got the parentheses up here, I put them down here. So everything makes a little bit more sense. So we are trying to add up to 100% of your DNA, all of the DNA, 10%, and A's, adenines, make up 10%. Now, what did Chargaff tell us? What was his rule? A's and T's are pairs. So if 10% is adenine, then what's thymine? That's right, 10%, because they're pairs. If you have 10 left shoes, you have 10 right shoes because they're pairs. All right, so let's take this 10% right here and we're going to move it, fill it up in there. You probably have a worksheet or something that goes along with this, so pay attention. So now we got 10%, 10%. That together, 10 plus 10, is 20%. So far, good, guys, right? Easy. Now, here is where it gets slightly less easy. I'm not going to call it hard. Slightly less easy. 20% plus something equals 100%. Oh my goodness, that is some hard math. 100% minus the 20 we know about. What's left? 80%. Easy, easy math, guys. All right? So 20 plus 80 equals 100%. Yay! But we weren't looking for that 80, right? This is C and G together. We want them separate. And they have to be the same number. How are we going to do that? Easy, guys, divide by 2. 80 divided by 2 is 40. 40% is C, 40% is G. Let's move those numbers in there. Cool. How hard was that? Really, really simple stuff, guys. And if that still confuses you, rewind. Now you have an assignment. Part of that assignment is going to be doing this again, but this time A equals 15%. And again, at the time of making this video, I did not make the assignment, so I don't know where this is going to be, if it's a Google Doc or a Google Form or what, but look on Google Classroom, and it should be there waiting for you. All right, ladies and gentlemen, that is it. Good luck, and I will see you in class.